So if you might remember, last time I was talking about the divisions of the nervous system, and now I want to talk about white and gray matter, what that means, and how it was that we came to understand white and gray matter and slowly began to understand the functional importance of this structural characteristic of the nervous system. So the first person we get to talk about is this guy with really impressive facial hair. That beard is something else. I just shaved my beard today, shaved all my hairs. Arcangelo Piccolomini, an Italian dude. Around the time of Vesalius, a little bit after Vesalius' death, he published an anatomical treatise called Anatomicae Praelectiones, explicantes mirificiam corporis humani fabricam. That's a mouthful, say that 10 times quickly backwards, in which he distinguished between the cortex and the white matter. He was able to say clearly, and this is at a point now where human dissection is becoming acceptable, and his description in his Anatomicae laid the groundwork for Malpighi and the work of van Leeuwenhoek that came after him. And then towards the end of his career, he became headmaster at Hogwarts. Sorry, dad joke there. Okay, this guy a little bit more dedicated to his to shaving. Van Leeuwenhoek, we've talked about him numerous times in terms of his microscopic anatomy. And he was the first person to really get a good depiction of a nerve fiber in cross-section. This is what we see here. And as you look at this, we've got, this is actually a pretty remarkable drawing. We've got muscle fiber along here with the fascicles. We have a nerve, and in modern parlance, we would call this epineurium. The connective tissue around the cut nerve, and then we have the individual nerve fibers. We see the dots would be the axon, and he didn't know all this at the time; he was just drawing it. But and then the white around the dot, the little donut shape, what is that? It's like little commercials where for car wax on, wax off, ax on. It's the axon and the myelin sheath. Yeah, that's the myelin sheath around it. And then the little black ring around that would be the endoneurium. But he didn't know any of that stuff at the time. He was just drawing pictures of what he saw. And it wasn't until our friend Robert Remack, who we've talked about before, I think I got choked up about the story about the discrimination that he faced as a Jew. His grandson died in Auschwitz, all that stuff, to remind you. In 1836, he, amongst his many other contributions that were stolen by Virchow and the others, he was able to describe myelinated and unmyelinated axons. And you can see in some of his drawings here, like in C, the panel C down on the bottom left-hand corner, you can see an axon with the what would be called Schwann cells wrapped around them. This guy, 2 years later, is like, hey, okay, Remack, that thing you discovered, maybe I'll put my name on it. You don't care, do you? Um, yeah, he wrote an account of the same cells in the peripheral nervous system and named the cell that wraps around the axon as a Schwann cell. Following closely on that, this was like a hot period, the late 30s into the 40s. This guy, Ballinger, he began to say, well, okay, this gray matter and the white matter, they're not just mutually exclusive. It's not like, a vanilla ice cream scoop here and a chocolate ice cream scoop there, and it's a little melty in between, but pretty much it's like two different flavor islands in your sundae cup, that they're actually connected somehow and that they are functionally dependent and related to one another. So here's a section of the cerebral cortex. The very outside of your brain would be at the top. You can kind of see the little bit of daylight up there at the top of that picture. And then there's this outer band and an inner band. These were described by him, this French guy, Ballinger, who those bands bear his name now, the outer and inner bands of Ballinger. All right. So about 20 years later, this person, Virchow, and we've talked about him as being, a German guy in an influential post in the academy in Germany there, and sort of being a clearinghouse for a lot of scientific submissions and ideas, many of which he would steal for himself and suppress the correct attribution. Well, he was able to take some of this work and coined the term neuroglia. And neuroglia to him, these were cells that were not neurons that acted as the glue that held the brain together. He also famously was quoted, omnis cellula a cellula. All cells come from cells. Everything sounds so much more official when you say it in Latin. Anyways, that tautology that seems kind of ridiculous and redundant nowadays was, at the time, not especially controversial, but a little bit so because cell theory was still, not a completely settled issue with every person. And you'll remember that Robert Remak had laid a lot of that groundwork 20 years earlier for understanding that cell theory. And then Virchow kind of snatched the idea, put it into Latin, and then collected the laurels, at least at the time. So because of all that, because of his plagiarism of Robert Remak and the suppression of the work of Ivar Sandström, the Swedish guy I talked about, I don't know, a couple weeks ago, I'm giving him the big L there. Sorry, buddy. That's what happens. All right. The next sort of leap forward in our understanding of the structural and functional significance of neurons and the relationship to the white and gray matter came from this guy Ranvier, another Frenchman, who was in 1878 was able to very clearly, building on the work of Robert Remak, was able to say, oh well, this myelination that we see, it gets interrupted in regular intervals. All right, these regular intervals, which to this day are often, they're still often called nodes of Ranvier, although more correctly are called internodes. Oh, all right. So, and I didn't really take the time to go through the functional significance of them, of saltatory and continuous propagation. But basically the idea is that the Schwann cell along the axon increases the insulation and conductance of the axon and allows the electric field to jump from one node to the other very rapidly. It dramatically increases the speed of transmission. And then each of the nodes is kind of like the transformer that you see every 10th telephone pole. You'll see those big garbage cans tied to like 1 in every 10 telephone poles. Because as the voltage drops, you step the voltage up by stepping the current down. And so you just pop the voltage up with those transformers every so often. And that's kind of what the nodes of Ranvier are doing. Yeah, because the nodes of Ranvier are where you have the voltage-gated channels that can open, and they don't open along where the myelin sheath is. So that was sort of the functional significance of all that. Let's look a little bit more closely with a cartoon at what we're talking about here. So here would be a peripheral nerve cell, and we have the soma, the cell body with the axon. And these Schwann cells get wrapped around the axon, and each segment is its own individual Schwann cell. There are other glial cells. So a glial cell, maybe I didn't make this point clear enough. There are neurons, and then there is this whole cast of other accessory cells, the neuroglia, right? Where were we here? No. It was this guy, Rudolf Virchow. The neuroglia that he sort of used to say, well, these are like the glue of the nervous system. The other glial cells that you'll find in the peripheral nervous system are the Schwann cells, of course, and then these things called satellite cells. And these are kind of like, I sort of think about the satellite cells like WALL-E. You guys ever seen that movie? The little robot, the cute little robot. It's like busy on its way, picking up garbage, making the world better, cleaning things, fixing things, wishing you had a friend. That's kind of like a satellite cell. They're just going around, they're buzzing around, fixing things up, taking off trash, monitoring the CO2 and oxygen level, the dissolved gas, and the neurotransmitter levels, et cetera, et cetera. They're kind of like the custodian, so to speak. All right, and these Schwann cells wrap themselves around the axon over and over and over, and their lipid bilayer, that lipid, there's a very high dielectric constant in the lipid, which means that it's able, or low dielectric, which means that it's able to be a good insulator for the electric field and keep the electric field that gets generated by the local current inside an axon during a depolarization, keep that electric field concentrated in the axoplasm. So what we see here is AX would be the axon or the axoplasm, and MIT is just a mitochondria, and MY would be the Schwann cell, the layers of you can see that striation, just like layer upon layer of the cell that just wraps itself around the axon. Hi.
So the Schwann cell itself is the myelination of the axon, or is it making the myelination? Like, are they the same thing? Okay.
Yeah, a Schwann cell is just like, it's just a cell that does literally nothing. There's like nothing in it. It just like, it just wraps itself around and around and around and around and around. It's what you would call a fat coating. Yep. Yep. Lipid coat. Yeah. It's literally just insulation on a wire. Concentrates the electric field in the center of the axon. They didn't really understand that in the 19th century, like what it was doing. They were able to describe it, and that was the first step towards understanding the functional significance. But like the the understanding of the electronic significance of the myelin sheath would come later, right? I mean, Emile Dubois-Raymond was, the first to really clearly characterize the speed of an action potential, or not even clearly, but get any kind of recognition of an action potential happening. So that was around the same time. They did not have the sort of physics in place to really understand everything that was going on here yet. But it's coming. Oh, just more pictures. Boy, I was having fun here with pictures. Yeah, well, here's a good one for you, Jay. You can see the axon, right, which is this thing. And these are microtubules. So cool. So cool. Oh, man, I love them, these microtubule arrays. The different tubulin monofilaments. And then the Schwann cell, this would be like the nucleus of a Schwann cell, or maybe a mitochondria or something there, who knows what it is. And then the rest of the cell is just empty. It's just like flattened out coils of itself wrapped around. And actually, Schwann cells are super important in regeneration because, nerves have a hard time regenerating, but it's not impossible, particularly in the periphery. I have I ground this thumb pretty much off, the end of it at least. I left the nail bed, but the whole end of it was gone in a sander when I was, probably like your age, something like that. That sucked. But it all came back. Look at that. It grew back, and I can feel all this. I have feeling here. And that's because as the tissue kind of grows back, the Schwann cell creates a little bit of a cone, it creates a cone, and the growing nerve fiber can go into that cone and grow into the pathway that the Schwann cell creates for it. Yep. In your lifetime, neural regeneration is going to be a thing. People with spinal cord injuries that can't walk and stuff like that, they're already making huge advances, but someday that is going to be a solved problem. I'll probably be gone. All right. In the central nervous system, the neuroglia, Virchow's neuroglia, are more numerous. So we have well, first, I guess we'll start with the ependymal cells because they're in the list here. An ependymal cell, these are cells that line the ventricles. So the ventricles, have I talked about the ventricles yet? Maybe, it might be coming up. Anyways, in the brain, there are, we looked at them a little bit yesterday in the VR thing, didn't we? They're like empty spaces. They're basically empty spaces that are full of cerebrospinal fluid. And they are lined with these things called ependymal cells that are producing and kind of monitoring and adjusting the cerebrospinal fluid. And then there are astrocytes and oligodendrocytes. Oligodendrocyte is, when you see that, think Schwann cell of the central nervous system. It's just like the Schwann cell of the central nervous system, except that they are not one segment, isn't just one cell. One cell can put out an arm that's one segment on one neuron and put out an arm that's another segment on a different neuron. And have its arms in all kinds of directions, insulating all sorts of different, maybe parallel, maybe not parallel axons. Likewise, astrocytes also have all these projections out. They're not neurons. They're glial cells. They put projections out and they surround the axons and the body of the neuron and create something called the blood-brain barrier. So it turns out that even though there's a rich blood supply to the head, that neurons themselves do not come into contact with blood. All right? They get their nourishment from cerebrospinal fluid, this sort of primordial ocean broth that's in your brain that the giant worm that is our central nervous system needs to float around in. And the rest of it is just a life support system for that, and helps maintain this cerebrospinal fluid broth. The astrocytes put up the wall that barricades the neurons away from the blood, because there's stuff in the blood that we do not want to get to the brain, right? And people eat all kinds of stuff. You get exposed to all kinds of stuff, right? The brain is delicate. It's very delicate. You do not want to expose it to all the crap that you randomly are running into in the day, right? So the blood-brain barrier helps with that up to a point. And then lastly are the microglia. And I kind of feel like the microglia is sort of like a satellite cell. They're just like, it's like these people driving around in little dump trucks or something like that, here to like pick up garbage or something like that. They just are sort of, yeah, again, they're like Wall-E of the brain, of the central nervous system. So why are oligodendrocytes different than Schwann cells in this regard, that they put out multiple projections to multiple different axons, do you think? You cogitate on that question. Think about it, and then I'll give you some more clues, and then we'll answer. I mean, the answer, I've kind of already told you the answer, but all right. Pío del Río Ortega was really the person that articulated all these different glial cells. And it was the person that clearly distinguished between the microglia and the oligodendroglia with a silver carbonate stain. Kind of a gifted scientist, but interestingly, he had, so I don't know how much you know about the history of fascism, but the Spanish Civil War, there were, fascists in power in Spain. Spain was in a bad way. Didn't really participate in World War II because it had its own problem with fascism that they didn't really want anything to do with the rest of the world. And there was a big civil war. The Spanish were standing up against fascism in their own country. The Spaniards didn't just fold like the population in Nazi Germany. They stood up and had a civil war, and this guy did not really think he was going to be good with a gun. Took off for Oxford, and then went to Argentina in 1940. Wanted to get as far away from Europe as he possibly could. But it kind of like interrupted his work. The science running into the cross current of history, sort of story. All right, so here is a picture of gray matter and white matter. Here in the gray matter, we have the cell bodies. That's why they're gray. And we can see ependymal cells. We can see astrocytes creating. So ependymal cells creating the cerebrospinal fluid. Astrocytes creating the blood-brain barrier, and also giving a structural framework for all these neurons. When you think about neurons in the brain, people have this idea that they're just like neurons all over the place and they're going every which way, and they are. It's very highly organized, but they are in like very highly controlled but also dynamic spatial relationship with one another. I ask you again, why are oligodendrocytes different than Schwann cells? Okay, I saw a knowing nod there. Give it to me.
No, I don't know. I was waiting for the answer.
Oh, you got it. You know the answer. You know it. Give it a shot. Just guess. Take any wild guess. It doesn't even matter.
to get to more places?
Give me, what does it mean?
Like the opposites?
Okay. It does have to do with spatial arrangement for sure. Anyone else want to elaborate further? Put the last few stitches in this sock before we gift it to the world. Okay. Imagine that you have, let me think about this. A bunch of straws. You got a bunch of straws, and you're trying to, you bundle them together. Yeah, I just saw that. So, like, you're bundling straws together. And in that bundle, the straws are all going in the same direction, right? Nah, it's not hard to bundle it together. You just wrap it up around the outside, don't you? But what happens when you are trying to build the Brooklyn Bridge out of straws? Right? Where straws are going in all kinds of different directions with respect to one another. You might need some struts that control that. Hey? So these oligodendrocytes are helping to maintain the spatial orientation of neurons that aren't all just bundled together going in one big tube all to the same place. Neurons are going all over the place in the brain. There's all kinds of places they need to go. And you'll have a bundle of them going in this direction. You have another bundle that's got to go right through that same direction. It's got to go through that same spot to some other place. And you need to control that. These oligodendrocytes are doing just that kind of thing, right? So they're able to coordinate sort of multidimensional constraints in terms of how different nerve fibers are growing in different relationship to one another. Does that make sense? Okay. So there are 6 regions in the brain. Erasistratus of Chios in Alexandria, he was one of the Alexandrians. We've talked about Erasistratus before. I've probably talked about Herophilus, his buddy, a little bit more often. But these were some of the only ancients that actually got down with some dissection of human cadavers because they were in Egypt, and Egypt was a little bit more enlightened in that regard. So in the year 280, he made a clear distinction between the different regions of the brain, the cerebrum and the cerebellum. And I'm pointing this out here because the sort of like intersectional story, the moral of the story that we're going to slowly unfold as we move forward here is related to scientific racism and the notion of how, the shape of a person's brain in either global sense or in smaller features was a proxy for intelligence or moral character or et cetera, et cetera, et cetera. This idea was not new. They did not invent this back in the 19th century, which is where a lot of it blew up. But this guy Erasistratus he thought that those convolutions that you see on the cortex up there, we call them gyri. A gyrus is one sort of fold, and the grooves in between them are called sulci or individual sulcus. He thought that those folds, that wrinkling on the surface of your brain, the more you have of that, the higher your intelligence is. So this notion was entirely rejected by Galen. He said that's complete bullshit. And we move forward. But when we get back to this idea, I don't want you to think that a bunch of like racist dudes from Great Britain came up with it. It had been kicking around for a long time. Hey, oh, hi. Yeah. So a fissure goes. It's like the tissue is not connected to one another. So like the Sylvan fissure or the median fissure, right? The median sagittal fissure, that is like the two hemispheres of the brain are next to each other, but they are not connected. A sulcus or a sulci it's just a divot. It's a groove, and if you go down into that groove, there's nothing but tissue. Like it doesn't go deeper than you see, but a fissure goes down. It goes much deeper, right? There's two. There's the central median fissure or sagittal fissure. Then there's the fissure beside the temporal lobe there. Maybe that's called the Sylvan fissure. I'm forgetting my anatomy right now. So anyways, later in the early modern era, Magnus Hund uses the word cerebellum, like little cerebrum, to describe the posterior portion of the brain in his book Anthropologia. We'll talk about him a little bit more later. Interesting character. And then the this guy Robert Todd was an Irishman, and he began to discuss the other parts of the brain. So the brainstem, the diencephalon, the hypothalamus and thalamus, the mesencephalon or midbrain, with the cerebral peduncles, the pons, and the medulla oblongata. He talked about the brainstem and then the associated basal nuclei, which are not a part of this. We looked at the basal nuclei in lab yesterday. You remember the corpus callosum and the amygdala and the putamen and the internal capsule, all those terms. Do you remember us talking about them maybe? Yeah, all of those structures would be on either side of this thing, but they've been kind of removed here in this image. But this guy talked about the importance of those structures in what we now call the limbic system, the emotional system, and in movement, emotion, etc., etc. Yeah, he talked about all this in a famous series of lectures called the Lumleian lectures, during which he would famously, to keep his, these lectures were epic. You think like I am long-winded. This guy would talk for like 6 hours straight in these lectures, and to keep himself going, he would drink cups of hot tea, strong black tea, with a little bit of whiskey poured into it, Irish whiskey, and it became known as a hot toddy, if you've ever heard that drink. But that's how he would lecture. I've always aspired, to that, but I've never actually come to class with a hot toddy. Yeah, kind of funny. Okay, so. I thought this would be a fun time, since we're talking about the regions of the brain, and emotion and the emotional cortex, and the frontal cortex. I wanted to tell you about this story about Phineas Gage. Has anybody here heard the story of Phineas Gage? Yeah, what do you know? Tell us the story.
He was like, he was like a mine worker or something.
Yep. I think he's holding it.
Yeah, and then like after he started to, his temperament was different.
Oh yeah. I mean, so this is what happened to him there. He was actually a pretty handsome guy for 1848. I'm sure he was quite a charmer, but then there was a blasting cap, and the rod, it took a metal rod and blew it up straight through. Oh, I should not laugh. Blew it straight up through his head, like up through his jaw, under his cheekbone, behind the left eye, and out the top of his head. Literally, like you see in the picture there, he lost his left eye. And here is they've done reconstructions, analysis of what the explosion must have been for Phineas Gage. So the thing that saved him is that he was like, "Ah!" His mouth was open when it was happening, and it just put it in the right angle so that the bar was able to get up under there and not completely blow his head back. So his head split, and apparently it temporarily hinged. His face and the back of his skull kind of like hinged. He had a fracture that went all the way across from one side to the other. The whole skull split in half and then hinged and came back together. And then the thing like blew out through the top of his head. What was interesting about this is that the dude didn't die. I mean, here he is, right? Guy didn't die. At first, people were like, "Oh, he's done." That's a hard one to come back from, but he didn't die. And the thing about it, he went from that like charming guy into a real jerk. He became a real jerk after that. Not just he messed up, but like he had personality changes, dramatic personality changes, and impulse control. So this is the moral of the story is study hard, don't become a minor, and protect your frontal lobe, because it's where your like impulse control, your higher executive function, and the coordination of like those inner thoughts that should probably remain inner for most people. That's your frontal lobe doing that for you. He lost it and had a very, very hard time in society after that. Yeah, so this was a really important medical finding at the time, actually, because it just illuminated what this part of the brain was doing for them. You know, the guy lived, and he had everything else was kind of normal for him. He could still see perfectly fine out of his one eye; they didn't get blown away, et cetera, et cetera. But the like sort of specific cognitive function of the frontal lobe was elucidated by this kind of horrendous accident. All right. So we're going to go now into the ventricles and cerebrospinal fluid and the blood-brain barrier. I'm kind of like pulling all of the pieces together. We're kind of going through neuroanatomy is what we're going through, and I'm putting the pieces together, and then we're gonna travel with those pieces later. Any thoughts about this? Questions? Yeah.
Is this what allowed people to realize that you could take out, like, do a partial lobotomy of the frontal lobe and be like, much better?
Yeah, certainly this raised awareness of the functional particulars of the brain. And, you know, you'll also see this is 1848. Keep that date in mind when we start talking about phrenology and, Galt and Broca and all these homies that are going to be so delightful to talk about, and how that timing works out. Because when I begin to show you like phrenologic maps of the brain, and like how different traits get associated with different parts of the brain, certainly this event, this widely appreciated sort of medical oddity, was something that played into how those maps and that understanding continued to evolve. Yeah. Good question. Anybody else? Okay. Next, we're going to go through all of these things: cerebral ventricles, CSF, and the blood-brain barrier, the triple B. All right. Back to our other Alexandrian buddy, Herophilus. He thought that certainly the brain is the seat of human intelligence, unlike Aristotle, you'll remember, who thought it lived in the heart. He thought it's in the brain, but specifically, he thought it was in the big holes in your head, the ventricles. There are these empty spaces, void spaces that are in the brain. He thought ventricles were the seat of human intelligence. Later, Galen jumped on board with Herophilus rather than Erasistratus, who thought the intelligence was in the cerebral gyri. Unfortunately, Galen's view becomes dogma. So you'll remember, what did they think? They understood there was a connection between nerves and muscles, because when a nerve gets cut, a muscle doesn't work. What was, thinking back to the muscle contraction theory, remember that? What was the muscle contraction theory that came from Galen and worked its way forward for a long time? Start with an H. Y. D. R. The hydraulic theory. Remember that nerves are pumping fluid into muscles and muscles expand, and it wasn't until they did the contraction of the frog muscle underwater that we dispelled the notion that the muscle fibers are changing in volume. Well, this hydraulic theory of nerve function, Herophilus thought that, and then Galen, as like the stamp of imprimatur from Galen, thought that this is where it starts. That hydraulic function starts in these fluid-filled chambers in the center of your brain, and when you decide to do something, it's that fluid-filled area that is like deciding to do it, and it's pumping fluid somewhere into the body to make a muscle expand or contract, etc. They thought we were basically giant hydraulic systems, and the ventricles were the source of all of that. Because they were trying to understand, how come there isn't blood in the brain? How come there is all this cerebrospinal fluid? They didn't call it that, but it was this like clear liquid that was in the brain. What's it doing? Well, they believed that it was part of nerve function and nerve contraction, and it was a result of this hydraulic theory. So this all makes sense to Galen, and it gets adopted rather than Erasistratus, who believed in the cerebral gyri, who was actually correct. That was the correct interpretation. But there's something to take heart here, eager young minds. That truth will win in the end, even if you are long gone and dead. All right, let's move on to the Islamic Golden Age and the medieval progress, if you want to call it that. So here's Avicenna. We've talked about this guy before when we were talking about muscles and nerves and whatever. He was sort of like the Galen of medicine of the Islamic age. Spent most of his time in hiding from the Twelver Shia controversy that was happening at the time. But he identified 3 ventricles in the brain. The anterior ventricle, which he thought was responsible for sensory processing and imagination. So still taking this Galenic thought that the ventricles are where the action is, and not all this other stuff around it. That the brain is just there to hold these little pouches of fluid at the center, and those pouches of empty fluid are where the action is. The middle ventricle is where your judgment and intellectual processes happen, and then the posterior ventricle is where memory happens. And I don't actually know why he cooked this up. I'm not exactly sure if there was any empirical correlate for these, or whether he just threw a dartboard at a list of cognitive functions and then scribbled them on his map of the ventricles. But all these anatomical insights, he is notable for at least clearly identifying and articulating which, of the ventricles there were. Instead of lateral ventricle, he called them both one anterior ventricle, and then a third ventricle, which is he called middle, and a posterior ventricle, which is what we call the fourth nowadays. Anyways, he took these anatomical insights and sort of like whipped them up into a special philosophical brew of mental function, and created a pretty complex hierarchical model. He did have a very detailed physical model of the brain's functioning and how that mapped onto these anatomical correlates. So that even though he was like wrong about them, that general notion of functional hierarchy, or we'll put it this way, structural hierarchy as being the foundation for functional cognitive hierarchy, that was a new articulation that he gave us. So that was kind of pushing it forward, even though all the details he had, at least in terms of the cognitive hierarchy and this sort of philosophical connection to the brain, were completely fictitious. And then, I got a typo there. 1509, our buddy Leonardo actually produces the first wax cast of an ox ventricle. That's fun. For the first time, really getting a good depiction of the shape of what the ventricles might look like. You can see this depiction here. This is from an ox brain. Human ventricles are a little bit different, but not wildly so. And then it was in the 20th century, in 1918 and 1919, where this guy, Edward Dandy, was an American who produced a mold of human ventricles and developed this technique called ventriculography or pneumoencephalography. Gotta say those slowly with braces. Ventriculography. I'll get these bad boys off soon. Pneumoencephalography. These were basically ways of visualizing the brain that they didn't have prior to this. So here's an example of a ventriculogram of a 3-year-old child that has meningitis, or an inflammation of the brain that's causing a distension of the ventricles. So like, woo, buddy, there's like you see how big those bad boys are? Yeah. So child's in sepsis, inflammation, overproduction of CSF, or a problem, probably actually a problem with resorption. He's probably got meningitis of the arachnoid mater. So they're not in the arachnoid granula, is not resorbing the cerebrospinal fluid. It's causing this sort of hydrocephaly in that child. Here's another one with a ventriculogram of a really distended ventricle in what's called communicating hydrocephalus. So communicating meaning that it's kind of blown out the walls between the two ventricles, so there's just one giant sort of cavernous space in the brain. And these are old pictures, right, from like 1920 or something like that. And you can appreciate by that picture, you're like, boy, is that light thing the brain? No, that is like the absence of brain. There's only so much space in your head. So this kid's ventricle is what's happening to his brain there. It's getting squished. Yeah, he's getting squished. Kind of a problem. But that ventriculogram was an important step forward in diagnostic technology. Here are what the cerebral ventricles look like. I do have a plastic mold of one in the lab. If you remind me next time we're in there, maybe during the heart dissection, I'll pull one out so you can see it. Yeah, there was, I forget the name of that book. There was some kid's book about it was about a ram that had these giant horns, and the horns were so big that he could ski. He could like climb up onto his own horns and ski down a mountain with his own horns. But it kind of what reminds me, the shape of the ventricle reminds me of the drawings in that little kid's book. You have these two giant horns that are the lateral ventricles, and then they communicate in the middle with this third ventricle, which kind of looks like the head of a moose or something like that. You have a little dot there in the middle for something that connects the thalamus, the two lobes of the thalamus on either side of the brain, through what's called the massa intermedia. And then that descends through the cerebral aqueduct down the middle of the brain to this fourth ventricle in the back, Avicenna's posterior ventricle. The tail of the fourth ventricle communicates directly into the central canal of the spinal cord, which runs the whole length of the spinal cord. So that's the ventricles of the brain. All of them are filled with cerebrospinal fluid, first described by Niccolò Massa, this Italian, in 1536. So in a very real way, our brains, like all of us, are like giant gray and white multi-tentacled worms that are floating in this huge flesh container of cerebrospinal fluid. And it has a number of functions. Buoyancy, right? So the brain is kind of floating there. It's how we are able to protect it from like getting slammed, right? And protects it, feeds it, gives it chemical stability in terms of nutrients, chemical messengers, hormones, and dissolved gases and waste products taken away. But this was the first description of it in 1536. And then our buddy Tom Willis. So we've talked about Tom Willis and the remarkable drawings that he made that really influenced our understanding of the brain. In 1664, in a book he titled Cerebri Anatome, just anatomy of the cerebrum, he suggests that it is produced by the choroid plexus, which was correct. That was actually an incredibly astute observation that Willis made. And at this point, people are still trying to come to terms with where the seat of the intelligence is, right? This Galenic notion, we still are dealing with the hydraulic theory of muscle function, the Galenic notion of that came from Herophilus of the intellect residing in the fluid of the ventricles is still kind of there. And what Tom Willis said is, well, okay, but this other stuff here, this brain, the stuff that's on the surface of the brain matter, is producing this fluid, so maybe it's not the seat of the intelligence. Maybe the brain, the thing that's making this stuff, is the seat. Anyways, he was the first to really describe the production through the choroid plexus and its resorption through the arachnoid granula. All of that is conducted by the meninges, and we talked about the meninges in lab yesterday, didn't we? We kept looking in the VR. We kept looking at dura mater. It's the durable mother, dura mater. You could all channel some durable mother here. It is a thick layer of meninges, of connective tissue that surrounds and protects the brain. In between the dura mater and the brain, the next layer down is what's called the arachnoid mater. And I don't know why I don't have a picture of it, but in this, I don't have a label on here. But in the, oh, I gotcha. I don't have the arachnoid mater here because Galen didn't name them. He named the dura mater and the pia mater. He didn't distinguish the arachnoid mater. He described the dura mater and the pia mater. Pia mater means little mother. So the durable mother and the little mother. The pia mater lays right on top of the brain. Later, this guy, much later, 1700 years later, Gerardus Blasius, a Dutchman with a sweet hairdo, in 1664 also discovered the arachnoid mater and called it such in recognition of the fact that it had like spidery, cobwebby lines that were a part of it. The arachnoid mater, do I go here? Yep. Later, another Dutchman, this guy Frederick Ruysch, confirmed that it comprised a complete layer surrounding the brain. The arachnoid mater has these structures called the arachnoid granula. Can you see my cursor up there? These arachnoid granula take cerebrospinal fluid that the brain is bathing in, and they return it to general circulation. This would be called the superior sagittal sinus. That it's a venous structure that's in the dura along the top of your brain. This is why injuries, that pierce the skull along the midline, along the sagittal suture, can be super bloody, right? Not a good place to get hit. But yeah, these arachnoid granula are how the cerebrospinal fluid goes back into the bloodstream. The same guy, Friedrich Reich, just since we're here, might as well take a minute to talk about him. He was also often called the artist of death. So he was an anatomical illustrator and anatomist that was kind of less concerned with really doing a detailed analysis of conventional anatomy and was more about sort of the sensationalism. And so I think this is ripe for critique here. He often did a lot of his dissections and drawings on infants, and we see him here with an infant making some sort of measurements. He was very fascinated with the relative proportions of, and one of the reasons he got fascinated in children was understanding the relative proportions. How is it that babies' brains are so much bigger than their bodies, and they're not that much smarter, right? So, like, this was kind of the question that was there. Like, what's the deal? Their brains are huge, their bodies are small and weird. They're not smarter than adults, and our brains are small compared to our bodies. So it's like he's trying to understand that scaling that was going on. And we have here him doing a cadaver on a baby that was stillborn with the placenta still attached, trying to identify what the umbilicus is actually doing, showing it going to the liver, et cetera, et cetera. And meanwhile, this like kind of freaked out looking kid here is holding an infant skeleton. He's like, whoa, what am I doing in this place? Yeah, just these. I'm going to show you some of the drawings that he made of these dissections, and hard to know what is real and what is not real in terms of his drawings. So here's another child with hydrocephalus that was probably stillborn, clinging to its placenta, or maybe it was alive, and whatever. Yeah, and often, yeah, his images were he was much more focused on artistic rendering and sort of sensationalistic artistic rendering, making a spectacle or a display, objectifying the human cadaver, the infants in this case. So really, but supposedly for scientific purposes, and taking advantage of the cachet that he might have had as a scientist to sort of gain fame through this sort of sensationalistic depictions of infants. And so here is, and he was trying to like situate these images in the natural order as well. So here he has all this like landscape. And it kind of looks like, what does that look like to you when you're looking at that? Huh? Yeah, he made these drawings. This kind of looks like, what does all this stuff look like back there? Looks like plants or something, doesn't it? Looks like arborizations. Yeah. Through his drawings, he was trying to find some sort of like reflection of the natural world inside the human body, but at the same time was sort of sensationalizing it. So here I'm going to show one more, and it wasn't just in drawings. He actually made a lot of these depictions, which were incredibly morbid. Yep, Friedrich Reich. Anyways, that was him. So, the arachnoid granula that Tom Willis initially hypothesized were described much more fully by Antonio Pacchioni, an Italian, in 1705 in one of his dissertations. And this is how we get the resorption. Later, about 60 years later, another Italian guy, Cotunga. I don't know how to say that. I've never heard it pronounced Cotuno. I don't know how to say it. Domenico F.A. Cotuno was an Italian that in the middle of his career was able to describe how this fluid is connected between the subarachnoid CSF that the brain is floating in and the fluid that's in the central canal of the ventricles. He was basically able to like get the plumbing of this whole system worked out. Meanwhile, much later, Max Lewandowski, German, in 1900, coined the term blood-brain barrier, the Bluthernschrank. Bluthernschrank. Does anyone speak German in here? I think it's called Bluthernschrank. He made the observation that you could inject certain things into people and expect it to have an effect on the brain. However, that didn't happen. And in particular, he did some initial work with bile salts. And this caused him to hypothesize that there was a blood-brain barrier. A decade later, this guy, Edwin Goldman, and Paul Ehrlich, in collaboration, find that this purported blood-brain barrier is impermeable to large molecules. So they were injecting aniline dyes, these colored dyes, into all sorts of animals. And although when you injected the dye into the animal, so it's a living animal, you're injecting aniline, which is not like a B vitamin or something like that. It's not good for you, but it would circulate around the animal, stain their tissue, and then they'd sacrifice the tissue and look at the staining that happened throughout the body of the animal. They noticed that the brain didn't stain in most species. Some species it did. And however, when you take the dye, specifically he used tryptin blue, which I think we used in class, when you just inject that straight into the central nervous system, the brain stained just as well as any other organ. But the brain wouldn't travel, it's overtime, out to any of the other organs. Anyways, that was how they initially established the blood-brain barrier. We'll pick up there with the structure of the PNS and sensory modalities.